morning, everyone. Thank you, Suzanne, Federica, and the panel, despite the technological challenges, for opening the forum with such powerful remarks and a fantastic discussion. My name is Chen Wan. I'm the Senior Research and Innovation Manager Health at GBC. With the real honor and privilege, I will be joined by a panel of stellar experts in this session, Exiting the Wait Room, a roadmap for how genomic tools can become a standard of practice in clinical care. Genomics and omics tests are no longer emerging technologies, but an integral part of patient care and the foundation of precision medicine. Despite the exciting technological advancement over the past 2 decades, and the many successes in bringing them into clinical care, challenges remain. They are often country and jurisdiction specific with shared commonalities. And this session, with their diverse background, our panelists will share perspectives on this topic and hope to shed some light on overcoming some of those challenges, collectively in BC and beyond. We are going to start with 3 short presentations, followed by the in-depth discussion, joined by the rest of the panel. We will also leave time for an interactive Q&A session with you, the audience. You can ask your questions anytime throughout the talks and the panel discussion using the Q&A panel on the side of your screen. You can also upvote questions as they appear. We will select the most popular questions during the Q&A session. Without further ado, I would now like to introduce the speakers. In the interest of time, I encourage you to check out our speakers' detailed bio on the Event Hub. Our first speaker is Dr. Yingtan Schrader. She is a clinical geneticist and the co-medical director for the Hereditary Cancer Program and BC Cancer. She holds a Tier 2 Canada Research Chair in Clinical Cancer Genetics and Genomics. Dr. Schrader is passionate about using innovative sequencing technology in research and clinical settings to benefit patients and their families. Our second speaker is Dr. Mari DeMarco. Dr. DeMarco is a clinical chemist at St. Paul's Hospital, the research director for Providence Healthcare, and a clinical associate professor in pathology and lab medicine at UBC. With expertise in protein biochemistry and laboratory medicine, Dr. DeMarco's research group works collaboratively with healthcare providers and patients to build biofluid diagnostics to fulfill unmet needs in medicine. Our last speaker before panel discussion is Mr. Craig Evany. Mr. Evany is the Chief Provincial Diagnostic Officer at PHSA. He is a laboratory services strategist with over 35 years of experience in Ontario, Alberta, and BC. He has been influencing healthcare through a series of senior level positions in different health organizations. He is also an adjunct professor, Faculty of Medicine at UBC. And with that, I'll hand it over to Yingtan.
Thank you so much, Chenwan. It's a pleasure to be here and be speaking. So I'll be speaking to you today on parent of origin-aware genomic analysis in hereditary cancer. As co-medical director of the program, we service the population of BC and Yukon in hereditary cancer genetic testing. So trying to identify a mutation in a family that may predispose to hereditary cancer and then offer testing to family members in order to give them access to ways to reduce their risk of developing cancer. I'd like to begin by the work that's conducted has been done on the traditional and ancestral and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh nations. I have the following financial disclosures that have been mitigated. Okay, so when we identify in the clinic a family with a hereditary cancer mutation, that means that there is that increased chance of developing cancer in that individual. There is usually a 50% chance that it was inherited from their mother or their father. And we tell that individual to tell their family members in order to get their family members in to have cascade genetic testing or genetic testing for that particular mutation, so they can then use that information to reduce their risks of developing cancer. So right now, the ability to know which side of the family it came from, the mutation was inherited from, is not possible in the clinic. And this is a day-to-day challenge that we face. So very recently, we've developed new technology, first in world technology here in BC, that enables us to determine which side of the family that particular variant comes from. And it's a combination of two different whole genome sequencing platforms that essentially combine different pieces of information that allow us to make this determination using only the blood sample of the child. So we published this recently in Cell Genomics, and this was important because all of a sudden it opened the door to different possibilities of how we could use this in the clinic. So in hereditary cancer, there's one particular syndrome. That a syndrome is just a pattern of features or cancers that we recognize. But when you have a mutation in the SDHD gene in particular, it can give rise to developing certain tumors. And it gives you a high lifetime risk of developing these tumors. In fact, if you inherit that mutation from the father, whereas if you inherit that mutation from the mother, you do not have that high lifetime risk of developing tumors. So this was a perfect use setting for this technology. And so what this means is that if you don't know if the mutation came from your mother or father, it means that we currently in the clinic tell you to do screening regardless. However, about half of those individuals may not actually need that screening. So we quickly went and identified individuals in the hereditary cancer clinic who had mutations in SDHD to see if we could show that this technology actually did what we thought it could do. And in testing so far, 18 individuals, we've been able to show that it accurately predicts the parent of origin every time in all 18 of those individuals so far. So where else can we use this important technology? Well, I mentioned to you right in the beginning that one of our goals is to actually offer genetic testing to family members. However, what we've realized is that genetic testing in family members coming in is actually poor. The rates are poor. And it's probably only about 30% of first-degree relatives actually come in to access this testing. We've found that also that the rates are lower in ethnic minority populations. And so it's an area that we could really improve if we potentially had new technologies. So here is a large pedigree. A pedigree is just a picture of a family tree. The circles are the females, the squares are the males. And what you see is a little arrow that points to one individual, and that particular individual has a hereditary cancer mutation. That individual's parents are deceased. And so it's hard to figure out what side of the family the mutation was inherited from because their parents are deceased. And so we tell that individual, we give them a family letter usually, and ask them to tell their family members to come in and offer to access genetic testing. And this usually takes a while, in the order of years, for all these individuals to come in. So what we could do with this new technology is actually be able to test that individual with the mutation and immediately direct our attention to one side of the family to actually say that it's one side of the family, in this case, the mother's side of the family, that is at risk for carrying that mutation and therefore at risk for hereditary cancers, whereas the father's side of the family, instead of being at 50% risk, is at closer to 0% risk for having that mutation and more like population risk. And they do not need to necessarily have cascade genetic testing. So this is important because it also changes the risks to carry these. And this is a completely new change or a paradigm shift in how we do things. But as you can see, it could be really important in terms of the speed of time it takes to identify individuals and also increasing their potential to actually uptake genetic testing, because all of a sudden it's less diluted, the risk, but it's more focused on one side of the family. So just to conclude by saying is that the prediction of parent of origin through this parent of origin aware genomic analysis is technically feasible in real world samples. It will be important for the management of patients with pathogenic variants or mutations in genes with parent of origin effects, like the SDHD example I showed you, especially when parents are unavailable or the parent of origin can otherwise not be determined. That's like in cases when individuals are adopted or the parents are deceased or declined genetic testing. It will enable immediate cancer risk assessment, and it stands to identify hereditary cancer patients faster, more efficiently, and at lower total costs. Thank you.
Thank you, Yingtan. Now we'll hear from Murray. Murray, over to you.
Thank you, Chen. All right, there we go. All right, thanks for the opportunity to share our experience in, not in genomics, but in proteomics. So a different type of omics and how we were able to translate some biomarkers into medical care. And we've done this over and over for multiple disease states and biomarkers, but today I'm going to talk about one particular program, that's the Alzheimer's disease biomarker program. And I'll start at the finish, which is we now here at St. Paul's Hospital in Vancouver, Canada, offer biomarker testing for Alzheimer's disease. It's a national program, so we provide this testing for patients, providers across Canada. Now I'll go back to the start. This is a decade-long story that I'm going to try and get through in 7 minutes, so bear with me. But from the very beginning, what we did is reached out to some key partners to talk about what they needed. So what were some of their challenges and what clinical questions could we help them answer? And from dementia specialists working in Canada, one of the key things they mentioned was the need for Alzheimer's disease biomarker testing. So they already had access to these biomarkers, these protein biomarkers, for research purposes. So if they were running a clinical trial, they could access these, but they couldn't use them in medical care. And then our other partner there in darker blue are the patients and family members who we also heard similar things from. They were also aware of biomarkers through clinical trials and other arenas, and they were asking us why they couldn't get testing, and we didn't have a very good response for that. So from the clinical laboratories perspective where I sit, our response was great. Let's look at what evidence is for their use and see how we move this forward, because there is a clear need here. The biomarkers, fortunately, were very well studied with decades of knowledge on these. We know that for a confirmatory diagnosis of Alzheimer's disease, we need evidence of these tau tangles and more specifically these amyloid beta plaques in the brain, but they leave behind a signature in cerebral spinal fluid. So this can help with early and accurate diagnosis, and they have high diagnostic accuracy for identifying Alzheimer's disease. They increase physicians' diagnostic accuracy when they add these to their clinical workflows. So that was well established. But there were some things that were missing. I'm going to talk briefly about the translational pipeline for biomarkers, and then some of our challenges around funding and technology and lacking some information about implementation in routine care. So from the biomarker pipeline, it's just very simplified view, okay? But you can kind of think of this as several mountains you have to climb to get to the finish line. So a new biomarker, that first stage of discovery, very exciting phase, right? A new biomarker's been studied, maybe in a small population. Then it moves on to testing in more diverse populations, and also testing by independent labs. And then finally over to this last hill you must climb, which is on the patient care side. And this is a little bit misleading because it looks a little bit more like this, where one side is the realm of academia and one is more the realm of healthcare. So that latter mountain, that's when you need the test that's ready for use in patient care, that has those appropriate characteristics, that ready to be deployed, it's safe for use, and testing in cohorts that you'd actually use that test for in care. And this is where my research lab sits and where my focus is also as a clinician for the hospital laboratory. We really focus on bridging this gap, as we ourselves have one foot on the research side and one foot in healthcare. For the Alzheimer's disease biomarkers, when we had those first conversations with physicians, we were sitting here. So this had really stalled out for years, if not more than a decade at this stage. So you can see how this could be frustrating to physicians. To span that gap, of course, we need funds to help us do the research we need to do. And so I've just put up some common criticisms and challenges we faced over the course of our efforts to get funding. And if we think about how funding agencies and programs could potentially help respond to these challenges. So one of the first things I note is, if a biomarker is ready for implementation in medical care, it's well known, right? You don't want something that was discovered yesterday deployed tomorrow in healthcare, but that lessens the novelty. And so that's something we heard. We also were shut out of funding programs because funding programs would say, we don't want to fund any research around amyloid beta because it's been known for a while. So we were completely shut out of particular funding programs. So that idea of balancing novelty and impact, I think, is important, where something further down the pipeline is less novel, but has much greater potential for impact. The next one that we heard from our scientific reviewers over and over again, so to paraphrase, there's no cure for Alzheimer's, therefore diagnosis isn't that important. And I hope to convince you otherwise based on what we learned through our work in the coming slides. There's also a misconception that biomarkers has to be commercialized to be of value. And we've demonstrated over and over again that this is not the case, and that we can deploy biomarkers in a cost-efficient and timely manner without a commercialization partner. So just a shout out to our funders so for their help, in particular the Alzheimer's Society and St. Paul's Foundation, which were really important sort of starting funders for this program. So technology, to climb that last mountain, one of the reasons this was stalled out was that there wasn't a technology that was appropriate for use in medical care. The technologies were okay for research. They weren't that great, but definitely not something you wanted to deploy clinically. So we first had to build the technology, and that's what we did here, is use clinical mass spectrometry to build the tests that we still use today as part of our national biomarker program. One of the other barriers was a lack of information about downstream effects of implementing these biomarkers. So to gather the necessary information, we launched this longitudinal observational trial of the use of these biomarkers. So we actually had to launch them into use in medical care, and we piloted it in select centers. And we looked at how these biomarkers changed medical decision-making. So how did physicians change how they were managing their patients? We also looked at health economics. So we're adding a test to the healthcare system, but maybe changing some downstream services or use of services. So how does the overall economics of this health system change with these biomarkers? And last but certainly not least, what is the value to patients and family members? So I'll just flashing up some major findings from the medical decision arm. Critically, it resulted in positive changes in medical management. So we saw an increase appropriate utilization of available pharmacological therapies. So increasing when appropriate, decreasing when not appropriate. And most critically on this slide, this decrease in additional costly diagnostic procedures, such as MRI or CT of the head, that physicians said they didn't need that repeat testing and that imaging testing, additional diagnostic workups, because with the biomarkers, they have a certain diagnosis. As far as a personal decision-making arm, I'm just showing some final quotes, a couple of representative quotes from patients and caregivers in our studies. So to answer those scientific reviewers who said, what's the value of these biomarkers and having a confident diagnosis? Well, our patients and care partners told us over and over again that this really helped them. Having a definitive label as to what was happening with their brain health was tremendously valuable. For care partners, it was particularly valuable in planning for the future and preparing for what's to come. And so I hope you've enjoyed this short story of 10 years of work. I just showed a few hurdles and how we've overcome them in launching the National Alzheimer's Disease Biomarker Testing Program. Thank you.
I really appreciate the unique and the complementary perspectives from our first two speakers. Now we'll hear from Craig. Craig, over to you.
Thanks, Jen. Good morning, everybody. So just want to quickly acknowledge that I'm joining from the unceded traditional and ancestral territories of the lands of the First Nations here in Vancouver, the Squamish, Tsleil-Waututh, and Musqueam First Nations, and I'm grateful to be able to live, work, and play in these beautiful surroundings. And before I go forward, I do want to provide a quick shout out to the two speakers who've preceded me with just amazing stories. I'm going to talk a little bit about PHSA, its mandate, and where we are in terms of advancing clinical genetics and genomic testing delivery in the province. This first slide just illustrates a mandate letter from the Ministry of Health directing PHSA to move forward with genetic and genomic testing and improve the efficiency and effectiveness of the priority clinical lab pathology service delivery areas. So what's interesting is that was the mandate that was presented to PHSA in 2019-2020. The story certainly goes back much further than that, but PLMS is the agency within PHSA that is accountable for driving that integrated approach to lab medicine in the province. And the focus for us when we look at all of our lab services, but in terms of focusing on genetics and genomics, the importance of having principles that drive an integrated approach. When we talk about an integrated approach, we probably need to step back and recognize that in British Columbia, there are 12 different autonomous lab organizations that operate labs in this province. 3 of those autonomous labs reside within PHSA. And all of those have their own mandates and their own funding streams and funding mechanisms. So it's the intention of PLMS and its predecessor was to, as the agency for pathology and lab medicine, was to act as the governance oversight for lab integration in the province. And that original mandate, which was established in 2015, was quietly set aside as more importance was placed on the accountability and ownership within each of the health authorities and lab system partners that provide services in the province. So as we move along, what PLMS has been doing since 2020 has been to build a target operating model that is intended to facilitate the planning and coordination of lab testing in the province, including genetics and genomics. Since the establishment of PLMS, and through the journey with COVID, we've had the opportunity to set up a much more interconnected relationship within the health system and all of the lab system partners, both on the private and the public side. We also strengthen our relationship with the Ministry of Health, and I think this lab target operating model is enabling us now to start to focus more squarely on how we advance important initiatives such as the introduction of genetics and genomics testing as we go forward. This organizational structure is intended to reflect on the complexity as well that we have in this province. And The agency does not have that overall governance nor overall funding mechanism to drive decision making and to coalesce resources. It has to look and rely on relationships and collaboration across the system. We are building, however, some interesting processes to enable us to move forward with genetic and genomic testing. We've established a provincial genetics genomics discipline committee. It has folks from a variety of different backgrounds at the table, including the Ministry of Health, including some of our leading folks in genetics and genomics. And through that process, now we have the ability to move through new markers for inclusion into genetic testing from a funding perspective. What we also have is a test review committee that we are reimagining. In the past, it was primarily focused on adding new testing to the MSP funding bucket, but as we go forward, we want to create a test review committee that actually looks at the value from a health system perspective, from the perspective of the patient, and from the perspective of the needs of clinicians. And through that process, then move forward and work with the Ministry of Health to identify the appropriate funding structures to support the introduction of new testing in the province. So these are some of the structures that we are currently building out. We're confident that we'll, in working with the Ministry of Health, will make a difference in the implementation of genetics genomics testing as we go forward. PLMS, by virtue of its role today, can play an integrator role that can help serve in terms of improving patient care, in terms of providing equitable access. We can align nicely with the 10-year cancer action plan and ideally move through a more responsive model than that of the past. I do want to say that the province, in terms of having 12 lab enterprises, also has a variety of different funding streams today for lab testing, some of which are global funding, some are fee-for-service funding. One is a contract for service funding, and some are specialty fundings that you find in terms of BC Cancer. The reality is that none of these funding methods today really address the dynamic needs of our genomics and genetics setting. And so I think there's a push from our perspective to work with the ministry and to work with our partners to seek out new funding models to ensure that we are more timely in having the right funding available to drive this in a British Columbia setting. And so I'll just close by saying that PLMS, from our perspective, my last slide is choosing not to advance. So our vision really is that we want to support a made in British Columbia clinical genetics and genomics testing environment that supports health system strategies, meets priority population needs, and drives investment in the provincial lab system. Thanks for your time.
That's great. Thank you, Craig, for the fantastic overview of PLMS. And it is now my pleasure to introduce the rest of our panel. Dr. Tanya Bubela is the Dean of Health Sciences and SFU. With her legal training and PhD in biology, Dr. Bubela's research program in intellectual property and health law focused on translational biomedical research and large collaborative science networks in genomics, gene therapy, and stem cell biology. Dr. Sterling Bryan is the health economist with extensive experience of engagement with health policy and decision-making worlds. In addition to his academic appointment with the School of Population and Public Health UBC, he is the Chief Scientific Officer at the Michael Smith Health Research BC. Ms. Carleen Todd is the Vice President, Access and Roche. She has 23 years of experience with Roche and held various roles in Australia and Canada. Ms. Todd is passionate about timely, sustainable, and equitable access to genomic testing in clinical practice to support the right patient receiving the right care at the right time. Last but not the least, Dr. Nathan Michaels is the Director of Precision Medicine and Genetic Services at the BC Ministry of Health. Dr. Michaels is a neuroimmunologist by training, and he has worked in a pharmaceutical policy and engagement environment for 3 years. In his current role at the Ministry of Health, he sets the strategic direction and leads the development of policy, legislative frameworks, and guidelines for precision medicine, gene and cellular therapies, and related innovative therapeutic areas for the province. And with that, we're going to move to the panel discussion. Let's ask the economist on the panel first. So starting, recognizing the value gains from like the healthcare investment. So how do we ensure the strong evidence on the effectiveness or maybe clinical utility and the cost effectiveness in the real world settings?
Thank you very much, Chen, and thank you very much for those inspirational presentations. And thank you for the opportunity to be on this panel. I'm going to start by just acknowledging that I'm joining you from the offices at Health Research BC, which are on the lands of the Musqueam, Squamish, and Tsleil-Waututh First Nations, the unceded lands of those nations. And also to say that I've got no conflicts to declare in relation to this topic, and that I work very closely with Dr. Janine Austin on this work in genetics, and that we always adopt a patient-oriented approach. So work very closely with those with lived experience of the conditions in question. So first of all, I think in answer to your question, I think we need an agreement on what represents value in a healthcare context. And I think having a broad sort of sense of being on the same page with clinical folks, with policy folks, as well as researchers and patients is critically important. So we talk about things like clinical utility, effectiveness, cost effectiveness, and patient values. And I think like if we can just sort of start off by being on that same page, I think that's important if we're going to make progress on implementation. And then I think we need to reflect on the analytic challenge in relation to that notion of value. So how do we get independent and robust evidence on whatever we have on that definition of value, clinical utility, effectiveness, cost effectiveness, etc. So I'm going to just very briefly tell you the story of the work we're doing in pharmacogenomics. And this is in relation to the chronic condition of major depression. Pharmacogenomics is work to select drugs and make sure that we're actually providing the most effective pharmaceuticals to individuals who live their lives with major depression. So the work we've done is, first of all, to understand the current clinical context for caring for major depression patients in British Columbia. And we've used administrative data to understand that. We've got clinical advisors and patients to help us understand that current care pathway, because if we're going to make change, we're going to make change to something, which is the current care delivery model. And we need to be reflecting on whether that change is a value proposition. We're in the rather fortuitous position of having quite a lot of randomized controlled trials that have been done in this area, and we want to understand what that evidence base looks like for the change that's being proposed, this pharmacogenomics change, introducing the routine use of pharmacogenomics in BC. And then we've built a simulation model. And this is what I describe as analytic infrastructure. So this is about mapping the current care pathways that we see. So we've got administrative data that helps us with that. And then integrating into that the evidence that we found from randomized trials as well as from other sources of evidence, so that we can actually model the current care delivery path. And we've got an idea of the cost. So for example, our best estimate is that the healthcare system in BC currently spends something like $9 billion on caring for patients with major depression over a 20-year time horizon. So we're able to make those statements and have some robustness to the evidence that we're drawing upon, we've also got evidence then for the effectiveness of the care that's being delivered. And then we can use that analytic infrastructure to understand the value proposition. So we're actually then going to sort of use the simulation model to say, what should the change be if we do introduce pharmacogenomics? And our best guess at the moment is that we can actually reduce the cost of care for major depression in BC by up to $1 billion over a 20-year period with this change. So I think the analytic infrastructure, having that in place, helps us in an iterative way to think through the implications of making change and introducing pharmacogenomics and working collaboratively with decision makers, with clinicians, with patients on how that might be. And one of the benefits, I think, is that it gives us the opportunity to ask what if questions. So we're always being asked, like, what if we actually don't do it this way, we do it a different way, we implement in a different way. It might be a different provider that takes the lead, for example, or it might be that we don't introduce it for the first time a patient receives a drug, but for the second time if they fail a drug, etc. We can actually address those questions using this analytic infrastructure. So I think building that analytic infrastructure that helps us inform policy decisions and implementation decisions is, for me, the way that we should be thinking about these questions. Thanks.
Thank you very much, Sterling. And the numbers definitely striking you shared. And so now I would like to probably ask Kerlin, from the industry perspective, like how genomics tests are being valued and evaluated by the payers and other decision makers.
Thank you, Chen. Thank you for this opportunity at Genome BC. And I just wanted to call out, I completely agree with Sterling's comment around the importance of value being considered and aligned across different stakeholders as well. And I apologize if there's a little bit of a delay with my video. I'm having a couple of technical issues here, but let's see how we go. So to your question, Chen, firstly, I'd like to acknowledge that valuing and evaluating genomic testing is challenging, but it's well worth our collective efforts for the benefits of patients and the healthcare systems. And ironically, some of these challenges in evaluating genomic profiling is actually due to the different types of value that these tests bring. And we've heard a bit of that in the stories that we heard at the beginning of the session. And so, for example, comprehensive genomic profiling in oncology has broad value as a universal companion diagnostic for numerous targeted therapies that may significantly improve health outcomes, offer smoother and more timely care journeys for patients, and more efficient use of healthcare resources. But unfortunately, a single diagnostic test linked to multiple potential therapeutic interventions is poorly suited to current HTA approaches that focus or tend to focus on a single test, single treatment pairing. And beyond the clinical benefit, genomic profiling also provides other types of value not currently considered in existing HTA value frameworks, including prognosis, the value of knowing that we've already heard about, the hope for patients and their relatives, as we heard from Intan regarding hereditary cases. Identifying patients who will not benefit from certain therapies is also really important in terms of reducing harm, as well as providing system benefits of increasing diagnostic accuracy, and also guiding patient participation in clinical trials more efficiently and more effectively to improve treatments for future patients. And participation in cancer clinical trials has been shown to be associated with improvements in survival. So all these benefits, the value of additional information, reducing the number of patients receiving suboptimal treatment, the benefits from increasing participation in clinical trials, are all important from a patient and health system perspective, but not currently considered in value frameworks. And then lastly, the value of genomic testing is dynamic, and it increases exponentially over time as we identify new biomarkers, new targeted treatments, new resistant mutations. And this brings challenges associated with the durability of value frameworks as well. And that's why CADIS 2023 watch list on precision medicines is really important. It's actually called out regulating and evaluating precision medicine technologies as one of the top 3 issues. And it calls out the meaningful impact of genomic testing in transforming health systems in Canada over the next 5 years. So while it's challenging to evaluate, it doesn't mean we shouldn't do it. And maybe if I could just add, a couple of policy proposals to value genomic testing and ensure that equitable access that we're seeking. One of them is looking at investment in genomic testing infrastructure, and decisions to access genomic profiling should be part of a broader healthcare strategy to focus on improvements in healthcare infrastructure. And interestingly, MRI has not been subject to conventional value assessment processes, and yet look at the infrastructure and the impact associated with MRIs. So decoupling the value assessment of diagnostic tests with the actual treatment itself would allow consideration of genomic testing as part of a broader investment decision in the long-term healthcare infrastructure, rather than continuing to link testing to individual treatments. And an example of where we've been able to do this jointly is to invest in genomic testing infrastructure in BC in a program called PREDICT, Precision Oncology Evidence Development and Cancer Treatment. And this initiative was built between Roche, BC Cancer and CFIN, and through shared investment and commitment, we've actually been able to bring earlier comprehensive genomic profiling to patients in BC at BC Cancer. And, what's exciting here is that, imagine if we could actually scale this model to provide genomic profiling to patients all across Canada by making similar infrastructure decisions in the same way that we have for MRI adoption. And then, in addition, policymakers should be looking at alternative funding mechanisms for genomic profiling to minimise delays in patient access. So well-designed programs to provide block funding and diagnostic services, exemplified by MRI and other medical imaging technologies, can help address those long-term sustainability challenges with funding as well. And another policy proposal is value-based healthcare, and this is where Roche believes a value-based approach to healthcare that does incentivise outcomes to patients and systems rather than volume of services delivered is essential for achieving sustainable and resilient systems. And 2 elements that are critical to that include having a systematic approach to evidence generation that we heard about from Sterling, and that includes real-world evidence relating to some of those indirect benefits and patient preferences for genomic profiling, and also considering what upfront investment is needed to realise some of those downstream cost savings. And so that requires breaking down some of those budget silos so that we can actually look at the long-term consequences. So with these recommendations in mind, I'd like to close with a little bit of a call to action. Unfortunately, the lack of fit-for-purpose value framework is slowing down access to diagnosis and genomic-driven personalised treatment for patients in Canada. So it is really important that together we rise to this challenge and we get it right for patients today and for tomorrow, because there actually is a cost of not doing anything, and that cost is high. So thank you again for the opportunity.
Thank you very much, Carleen, actually, for bringing a lot of really, really good points and perspectives. And in particular, really appreciate your speaking and starting from the patient perspectives. And you actually already touched on and perhaps already answered one of the follow-up questions I had in my list to really make my life easier. I would like to circle back probably a little bit to Mari. And you have extensive experience in clinical analytics, clinical validation, and you heard from Sterling, from Carleen. So I just would like to ask you, what advice do you have for the technology developers or either researchers or clinicians trying to develop those tests to meet patients' needs?
Lots of advice. I know you asked us to keep our answers to 2 to 3 minutes so that we could have time for audience questions, but I'll maybe try and capture a few key points of that. And, Sterling and Carleen addressed some important factors for, kind of later on in the pathway. So maybe I'll pivot and talk to, a little bit earlier in the pathway of someone maybe you've identified a biomarker, or maybe you have a technology that you think has promise. What do you do to help ensure maybe longer term success of that project? And I think you heard a lot of us kind of mention, having the key people at the table, and maybe you don't think that they're important to have, at that very early stage, but I would encourage you to involve the partners. So I think everyone's mentioned patients and care partners. Of course, the physician groups who might be ordering that testing is critical, as well as the people who would be running that testing. So if it's something that's going to be deployed in a clinical lab, having a clinical laboratorian as part of your team, even in an advisory capacity early in the project, can be tremendously helpful in, sparing you going down unproductive pathways. So the same as, if you were developing a new surgical tool that a surgeon would be using, you would definitely have a surgeon on your team, right? So if you're deploying a new clinical test that's going to be run by the clinical laboratory, you definitely want some clinical laboratorians on your team. And they can advise on things like even technology selection, like what is likely to be, appropriate platforms and such that will help your biomarker transit through this pipeline faster, can advise on some of the regulatory things you might face later on and how you can kind of prepare for that earlier in the pipeline. Even in biomarker selection, which ones are likely to meet the standards and qualities that you're going to need later on the pathway. So that really help you in those selections that will ensure success later on. So we've had those opportunities to be involved in early discussions with various projects that people are running and help try to provide those insights early on. We've also been involved quite late, and we can in various projects, and we could see how a lot of time and energy possibly could have been saved if those interactions had happened earlier. So I'll just wrap it up there in the interest in time, but I say assemble a team of people that will have some stake in the project through to the end, through to implementation.
Thank you very much, Marie. Great point on engaging, the potential partners later on upfront, and also looking to the regulatory landscapes down the road. So with that, I would like to switch to the lawyer on the panel. So Tanya, keep in mind and also recognizing the broad and very different genomic and omics tests may fit into the regulatory landscapes. And what would you like to highlight to the technology developers like Mari and others, as well as decision-making bodies, to pay attention to, and maybe perhaps to think ahead?
Yeah, thank you. And thank you very much for the invitation. And I'm coming to you from beautiful, sunny, unceded territories of the Tsleil-Waututh, the Musqueam, and the Squamish First Nations. And thank you for the opportunity to be here. So I think that many of the points that I would have made have been made. Mari was absolutely bang on in her comments just now on making sure that the implementation team and the case-making team is part of your technology development from the get-go. So there are multiple ways that you want to think about your early stage technology development. First of all, I'm assuming that you're trying to meet an unmet need within a system, whether that is to improve patient diagnostic, their journey or their diagnostic odyssey through the system. So as an example, we did some work with mitochondrial diseases that have systemic effects and are notoriously difficult to diagnose. And so looking at what would the impact on the cost savings to the health system be if those patients received a genetic diagnosis, a clinical diagnosis earlier through their diagnostic odyssey. So that points to Brian, just Sterling's comments on value, and so so having patients and clinicians and having the lab tech people in as part of your team is essential. So starting with thinking about implementation. Now, when you start to think about implementation, it will inform your development trajectory. and it will also inform the legal pathways that you need to consider in developing your technologies. And so here there are a couple of things at play. The first piece is the different commercialization or translational models that you want to take into account. And for genetic tests, as an example, there are a number of different commercialization models, some of them which require a commercial partner and some of them which require a health system partner, because of course within a health system, the health system itself is in fact the major market for the tests that you are developing. So most commonly, genetic tests are developed as laboratory developed tests. And these often obtain a pre-market clearance from a regulatory authority to market the test as a diagnostic kit or a companion diagnostic. That's another model. Develop the market for a diagnostic algorithm as software as a medical device. You can sell one or more of components of the diagnostic test as an analyte-specific reagent, or you can provide the test for investigational use only. The majority of tests in this space are laboratory developed tests, which do not necessarily require going through a national level regulator such as Health Canada or the Food and Drug Administration in the United States. Instead, what is regulated is the laboratory. In Canada, those regulations are largely provincial. In the United States, they can be under Clinical Laboratories Improvement Amendments, so CLIA certified laboratories, or they can be state licensed laboratories. When you're thinking about the intellectual property protection that goes alongside those different commercialization models through the different regulatory forms, what you really want to be considering is whether there is a market that exists or whether you're looking at a very rare or a neglected condition. So there's usually much less investment that is required for laboratory developed tests because you're working with the implementers as you bring that technology through to implementation. And it can be performed in any accredited labs, but you can't then commercialize it and sell it as a kit for use in other laboratories. If you do think that you have a system that you actually would like to develop into a full-blown device and market that as a diagnostic kit for use in other laboratories, then that will come with a much greater need for venture capital or investment. Because meeting the regulatory standards takes a lot of investment in the tens and possibly even the hundreds of millions of dollars, because you're going to need to run trials for clinical validity. And generally, hand in glove with that level of commercialization comes the need for intellectual property protection of different kinds. And there, it's really a strategic decision about the length of time to market as to when and what you might want to seek intellectual property like patents for, and what you will keep as trade secret. The software component is really quite interesting. It is under regulatory reform right now, which is the actual regulation of the algorithm that is the computational arm of the actual clinical test. And that can be regulated separately, as where they're actually regulating the software. And regulators around the world are kind of scratching their heads and trying to work their way through protecting or thinking about software. I think that's going to become a much bigger issue as we move into, for example, machine learning algorithms for example, image diagnosis. How you're going to, in fact, regulate those algorithms that are continually improving over time as they learn from more and more data is a challenge for regulators internationally. The final thing that I'll just say is about companion diagnostics. This is an area that is a real challenge for regulators because the companion diagnostic has to be part of the therapeutic that it's directing. It has to be part of the labeling. And so from a commercialization perspective, that can be quite challenging. It goes through much more seamlessly when you have the same company, such as Roche, that is developing both the diagnostic and the therapeutic simultaneously. It's a lot harder to crack into that market if you're just developing the companion diagnostic, and the therapeutic is being developed elsewhere because it can impact the market. Thank you.
Thank you, Tanya. What a great overview of this really, really big topic, and really appreciate you mentioning the upcoming challenges like software reforming, regulatory, as well as when we get into the AI imaging diagnostic age. And I think the topic itself would worth a panel discussion. So for the interest of time, I'm going to switch to Yingtan, and you're at a very unique position. You work with the technology development team very, very closely, and you're also the adopter and the receptor of the technology because you can basically implement this innovative test in your clinic to immediately benefit the patients and their families. So can you maybe highlight on the challenges, additional challenges and concerns that hasn't been touched on yet by the panel, but keep you awake at night?
Yeah, great question. So being, I guess, in this position of being able to see the clinical need is absolutely lights a fire under you in terms of trying to say, how fast can we get this implemented and benefiting the patients and families that truly would get an extreme amount of utility in it. And I know firsthand from being in the clinic, it's the type of technology that we could use every single day to help with variant curation, to trying to figure out the pathogenicity of a mutation, trying to do exactly that is understand what side of the family the mutation came from, and also just helping in terms of that actual patient management in the example of the SDHD that I gave earlier. So this is, the need is absolutely there. And the question is how can you implement something that is such a paradigm shift? Because truly this does not exist right now in clinic, and it's very novel in the concept that you could test a blood sample of a child or the patient at hand, and this could be a child that's 60 years old, where their parents may be deceased, and you need to know what side of the family to go after. You need that information in order to direct their management. How do you educate patients? Yes, but providers, like this is a real change in what's available into the actual technology. It's both elements of the combination of the two platforms I mentioned earlier. Neither of them are in clinical use at the moment. They're early adopters around know about these types of technology. However, they haven't become sort of mainstream in their use even individually in, I think, the research spheres. So it's bringing it into this idea into the mainstream of the clinicians who would be offering this test, that the technology allows you to do this now, and what are the potential implications of it. And so so the important things are education. What keeps me up at night is basically, how do we get this in fast enough? How do we get people educated on what it is and what the potential utilities are? But what are the hurdles? If we all of a sudden can test one person and then predict with almost 100% certainty what parent carries that particular mutation that's identified in that person, there are certain ethical questions that come up. And, those come up in the minds of clinicians immediately is, do the parents need to consent? These are not things that haven't already been encountered in clinical genetics. And so We offer cascade, or we offer family member testing, and sometimes test out of order. We might test a cousin first before we test the intervening parent, and then the uncle, just say, and that, you know, by learning that that cousin may have a positive mutation, it infers the status of those individuals in between, that they also have positive mutations. So we do that in clinic currently, and it's because we, you know, take the principle that even though we don't have the consent of those individuals in between, it's important that the person at hand has come to the attention and they've made that autonomous decision to get genetic testing themselves. It's just thinking about what happens if it's at greater scale, basically, in terms of these. Another example is we test identical twins, you know, and we don't necessarily get the twins' consent if we want to learn the genetic mutation status of the twin at hand. So in thinking about this new technology, I think what we're doing is trying to decide what are the focus groups that we're going to need to lead and have with other providers and professionals to understand what their potential concerns would be, what sorts of education they'll need is going to be important in there. We have an ethicist involved early on, and that's Dr. Alice Varani, who's director of the PHSA Clinical Ethics Service, to be able to help guide us in terms of some of these questions. The other piece that's really interesting about this technology is that for the SDHD example, where you would actually change someone's management, it absolutely, I would say, needs to be clinically accredited, or you need to, you know, it needs to go through those sort of rigorous processes to understand that it's giving you the right answer. Whereas the application of actually directing the cascade genetic testing, well, that's very interesting, because that's something where even as a research test, all you're doing is predicting which side of the family the mutation came from. It's actually even valuable if it was even as a research test, because you are internally, I guess, validating it, because you already know the mutation of that individual. And so when you run the test, you'll see that mutation and confirm that you've tested the right sample. But then you're just predicting what side of the family the mutation came from. And so in that setting, you're doing a second clinical test, and you're offering it to that individual where you predicted which side to reach out to first, and it'll be clinically confirmed. And so it's interesting because there's real benefits that can be immediately implemented, even if it was a research test. And so it's educating around that and running that actually on a research trial to see if that's possible, but not taking away from clinical standard of care. Like genetic testing would still be available to the other side. I think those are the main pieces, I would say, is that it's just about that educating early on. And my acknowledgement slide, I didn't put it up earlier, but you know, this technology came out of the labs of Dr. Peter Lansdorp, Dr. Steve Jones, and they're talented students as well. And at every opportunity, we've been trying to knowledge disseminate, you know, go to meetings. I'm in a meeting currently right now, embed it in the med school education, in graduate studies education, so people understand that this new technology is out and available. You know, hand over my slides it's being showcased at a course in Stanford right now, basically, just to essentially get the message out on something that is very new and hasn't before been contemplated that it was possible.
Thank you very much, Yingtan, for highlighting the ethical challenges as well as the importance of educate health providers. So that's also gave me an opportunity actually to call out, give a shout out to our next session, which will have a focused discussion on genomics education for health providers. With that, I'm going to switch to Craig. Craig, I think you touched on this a little bit in your presentation already. Like after hearing the panel discussion, I'm wondering if you can maybe dive into a little bit of context of a PLMS role in this whole process, and perhaps also the mandates that fall out of a PLMS, but very important for other stakeholders to understand.
Yeah, thanks, Chen. And you know, PLMS is an offshoot of the initial creation of the Agency for Pathology and Lab Medicine. And as I said briefly in my remarks, the mandate has shifted away from that critical role of governance and oversight of governance and funding of lab systems in the province. So governance of a lab system, and there are 12 lab enterprises in the province, remains with those lab enterprises themselves. And the funding channels that we have in place in this province remain directly linked to those labs. So if you look at a lab within a health authority, their funding channels are built into the global funding mechanisms. If they have some specialty testing, they may have some special funds afforded them, and they also may do some community-based testing, so may access fee-for-service funding. So the reality is that lab funding in the province has a lot of context historically in terms of how lab tests are funded, perhaps much differently than how infrastructure associated with MRIs was funded in this province and other jurisdictions. And that creates a lot of challenges and hurdles in terms of thinking about a provincial perspective on structuring a lab operating system across a provincial network. So if you contrast that to Alberta, where you have a single authority, Alberta Public Labs or Alberta Precision Labs as they're named today, they have the ability under their mandate to actually structure funding to move with where the needs need to be placed more specifically than in the British Columbia environment. And I would say that the role of PLMS then is to actually drive engagement with the lab providers, with the Ministry of Health, drive engagement with the clinicians in the lab space, but also through them to connect with the clinicians who identify clinical need. As it relates to genetics and genomics testing, what we've heard is that it's much more than just purely the sequencing of a gene or the mechanisms of using equipment and testing, but it's the entire infrastructure that wraps around it that does not fit or lend itself to the current funding infrastructures that are leveraged for lab services in the province. And so in many ways, it's thinking about lab services as an investment, thinking about lab services in terms of the upstream to the downstream value that we've heard about. And so in our role, ideally, we see ourselves as working hard to bring together the core partners to actually support and drive changes to some of those mechanisms so that we're able to more rapidly deploy and drive new testing into our system, but also then have the mechanisms to dial down on or turn off other testing as its utility may not necessarily be required in the future. So, we're building those infrastructures through PLMS to be able to support a much broader network. And the original intent through previous mandates from government, if you go back 2018 and before, there was a much more broad thought process into what the agency might be able to guide for the province. And we've had to make our adjustments and recognize that it's really about relationships and partnerships, but also trying to tear down some of the barriers associated with funding regulation and funding legislation that have put lab testing into very specific silos. So it's a really privileged position that I have to connect with so many folks across the entirety of the health system and to learn and work closely with clinicians who are striving to bring forward new testing and new new markers in the genomic space, but also in all aspects of lab medicine. But the reality of it is that we stand to benefit by working very closely with the Ministry of Health in a partnership model. Stand to benefit by working closely with all of our autonomous lab providers in the province to seek out solutions and to really ensure that the voice of lab is heard as we become a partner in planning for some of these broader contextual changes that are necessary to introduce some of these innovative approaches to lab medicine that are born out of lab medicine and born out of genetics and genomics. Chen, I hope that helps with your question.
Yes, thank you very much, for highlighting the uniqueness of BC system as well as different funding mechanisms. So Nathan, I think people has been waiting to hear from you. And I do have an unscripted question, which is asking you to address how, from Ministry of Health perspective, how should we effectively manage the tension between the scientific evidence and the economics? But as the last one to speak from the panel, please feel free to address anything you would like to bring up from hearing all the discussions.
Sure. So yes, the good fortune of being one of the last panelists to speak, I feel like I'm going to try add to the discussion rather than just summarize a lot of the points that have been made already. But that is kind of encouraging at the same time that we can have so many different panelists representing so many different areas of the healthcare system and seemingly in pretty broad agreement about what needs to happen next to really drive some effective change. But to answer your question, I think we need to recognize here that we are working with a market that has a lot of potential for really exponential growth. Depending on the reports you read, depending on base year used, the parameters that are included into market projections, the global genomic testing market is going to grow. If your base case is 15 million, which in some reports it is, they have it going up to 15 billion in the next 10 years. Some projections are a lot larger than that. But the kind of overarching theme is that you're looking at compound annual growth of about between 10 and 15%. Undoubtedly, we're not going to be able to supply the monetary resources that are going to be able to keep up with that growth. So naturally, because there is that disparity and that disparity is increasing, that creates a tension there between an ability to pay for new technologies and new science and what's available for our clinicians and for our patients. So in light of that tension, I think the question is, how do we maintain being able to provide patients with optimal care and being able to implement new tests and in an equitable and efficient way? And this is largely where I'm going to kind of reiterate some points that were already made by my other panelists, but I'm also going to try add a few things here. But I'll touch on a few things. And the first one, and I feel like it's the foundation of everything else that comes afterwards, is those really strong networks and those really strong avenues of communication between all those different partners within the healthcare system. So the Ministry of Health, the health authorities, our researchers, our clinicians, our patients, our patient groups, our private entities. When there is a collective and good information sharing between all those different parts of the ecosystem, we have a better ability to identify our common goals. And I think my fellow panelists would recognize or agree with me that we all are shooting towards the same thing. Another benefit there is just our ability to understand the perspectives of everyone else that is working in potentially a little bit different of a microenvironment. Someone working in a health authority versus a clinician versus myself at the Ministry of Health. We want the same thing, but we might have different challenges in our respective roles. So being open and frank with discussions and working together as much as possible, we can kind of understand those respective challenges, understand where we're both or all trying to get to, and then come up with plans that kind of take into account all those different factors. And then born as a byproduct of that good collective kind of vision and working together, more effective resource planning. Ideally, it's done as upstream as possible, involving all of your different stakeholders at the earliest stages possible. I know we've heard today from some of the great talks that started the session. There's more than just a test when it's being implemented. There's the associated things that come alongside it, the equipment that's needed, the reagents, the human resource requirements, the genetic counseling needs in the province, clinical geneticists, lab technicians, the informatics requirements, or the data infrastructure required, the privacy, ethical, legal requirements, policy requirements. I mean, those are things that if you can start working on them a year in advance or 2 years in advance, 6 months in advance, as early as possible, then it reduces that time and kind of reduces some of those barriers between a test being identified as useful and when it's actually implemented and available for patients and caregivers to use. Because obviously we can't fix those challenges within months. Policy changes, legislative changes require some time. And then another part, and I think a really crucial part, is good HTA, good evaluation processes, good management of entry and access of different technologies into the system. It comes down to just paying for what provides the most value to the healthcare system. And we had Carleen and Sterling talk about what value actually means, and having a clear definition of that is essential as well. But managing access and exit, paying for what works, not paying or continuing to pay for what doesn't, maybe reassessing what tests are in the system at a certain time point so that we can kind of ensure that we're always providing optimal access to what the best things that are available. And then I know we have quite a bit of, we're running quite short on time, so I'll just briefly mention too, something that Craig has talked about already, is just the better financing approaches for genetic testing in the province, more agile strategies, nimbler financing approaches. I'm not going to say too much more than that because Craig already touched on it quite a bit. But those are just a few things that I think are absolutely essential to managing that disparity between an ability to pay and what's available. And I guess I'll turn it back to Jen.
Thank you, Nathan, for the additional insights and pretty good summary of the panel discussion. And again, made my life very easy. So I'm going to take a couple questions from the audience now. I think one of the most popular question I think is for Intan. So our audience is asking, do you see parent of origin testing becoming standard of care for all hereditary cancer genetic tests? How would this change genetic counseling approaches? Thank you.
Yeah, great question. I think this comes to that part of the need for consultation with the genetic counselors and with the professionals who would be delivering this. Yeah, I see the future as this. I see the future of this as standard of care for all genetic conditions. I mean, what I didn't get into in the presentation is that we can understand parent of origin for all autosomal variants, so on every chromosome, with that same certainty. And so it goes actually beyond hereditary cancer. You could think of it in cardiac genetics and in all other aspects. So with that, how do you introduce a paradigm shift? It's with, consultation and with consideration of what's needed in terms of those pieces around education. I do think that all of the elements of genetic counseling will need to take into account, if this does become, standard of care in future, we'll need to take into account those pieces of, the prediction of parent of origin And understanding that it is just a prediction. We know that variants or mutations can occur de novo, or they can occur just in that individual alone. We know that there's non-paternity. There's other elements that will need to be, considered as well. And we will need to work as a team and work collectively with professional organizations and, with patients. I mean, this is going to be critical. And I didn't mention this as part of this, but we need to have the patient focus groups, and we need to understand, what pieces of information they need up front in order to feel like they made an informed decision about undertaking that testing and what it means, in terms of potential implications for how they're going to communicate that to family members. There is going to be implications on and consideration of talking to ethics and legal as well. I mean, there's literature around duty to warn. Like if we have some certainty over certain variants or certainty over prediction of who may carry it, then at what level of risk does there need to be a conversation around, we should really warn this individual that they might carry this. And so that goes into the secondary finding conversation. This is a genome-wide technology. It can be constrained, I should say that. The idea of doing like virtual panels on a genome backbone where you might hide some information, but all of these things need to be considered, and they will all need the consultation basically with the various stakeholders who are going to be doing this.
Great, thank you, Yingtan. So I will try to take one more question from our audience before our time ran out. I think this question is to Sterling. How far along in GBC in building the analytic infrastructure that you mentioned? If you don't mind, briefly comment on that, that would be great.
Sure. So we are in a position where we do have a model for major depression for British Columbia, which maps the care pathways that we currently see and gives us the opportunity to think about changes to those care pathways, be they genomic changes, or maybe this is heresy to say it, but perhaps non-genomic changes to care pathways that can improve care delivery and quality of life and the cost of care for patients with major depression. So it's up and running, and we have it, and we would be happy to talk to folks who have an interest in improving care delivery for patients with major depression and using our analytic infrastructure.
That is great, Sterling. Perhaps we should also highlight there is a really cool animation that's actually being made to illustrate and explain this micro simulation model, which is actually up on YouTube. We can definitely share the links with our audience. Okay, so with that, I'm going to wrap up the session. First of all, thank you very much, the audience, for the engagement and all the great questions. And we also thank our panelists for their time and enthusing and passionate and very invaluable insights. Very unfortunately, we are not able to ask all the questions, and we would love to continue the conversation with you offline and today's in-person reception if you can make it, or other occasions. We also hope this is not the end of this conversation, but rather the start of many important ones down the road. More importantly, we hope to have planted some seeds for the future partnerships that lead to the collective effort shifting the paradigm so people in BC and beyond, thus including all of us, can benefit from those innovative genomics, omics technologies and precision medicine. This concludes our session. We hope you have enjoyed the conversation. We will now have a lunch break, and our next session starts at 1:15. You can access that session by returning to the event hub and clicking on the next session. Thank you.